Hello students, a warm welcome to all of you in ITLS Academy and also welcome to all in our 6-month online certification course on topic Artificial Intelligence and Molecular Biology. In today's lecture, we will discuss about the model organisms. What is the model organisms and what is the role of model organisms in molecular biology. What is a model organism? Model organisms are those organisms too. What is a model organism? Model organisms are those organisms which are used in research field. These model organisms are used to understand the different biological processes or phenomena. Okay, what is a model organisms? Model organisms are those organisms which are used in research field to understand the biological phenomena, and the discovery of biological phenomena in model organisms is applicable to other organisms also. And these model organisms play the most important role in molecular biology and genetics. Model organisms or model species are widely used in molecular biology and genetics. Why these model organisms play the most important role in molecular biology and genetics? Because they are easy to maintain, reproduce quickly, and have a genetic similarity with more complex organisms. Suppose we want to study the genetic inheritance pattern in human. Okay, and this human is very complex organism. This is very complex species. The study of genetic inheritance in humans is very tough task. Okay, that's why here we used model organisms. Suppose here we use mice. Mice is a model organism to study the genetic inheritance pattern of humans. Okay, this why we use this mice? Because these mice have a genetic similarity with humans. And these biological processes may be gene function, disease mechanisms, etc. Okay, why we use this model organisms? Because we does not why we use this model organism? Because we does not perform a every discovery in complex organisms. That's why we use model organisms, and these model organisms are easy to maintain, reproduce quickly, and have a genetic similarity with our complex organisms. Okay, and these organisms serves as a powerful tool to understand the biological process. Now we understand the importance of model organisms. First is simpler system. Many model organisms have simpler genomes, short lifespan than humans, and making them easy to study. For example, if we want to understand the biological process of human, then we use mice as a model organisms. The genome is model organisms is simpler than the humans, and the lifespan of mice is also short as compared to humans. Ethical consideration. Research on model organisms can often be conducted more ethically than on humans. Okay. Next, conservation of biological processes. Many functional biological processes are conserved across the different species, meaning that discovery made in model organisms can often be applied to humans. Okay, there are lots of biological processes are conserved. Okay, whenever we discover these processes on model organisms, then these discoveries also applicable on complex organisms such as humans. Okay, that's why these model organisms play the most important role in research field related to the complex organisms. And the example of complex organisms is human. Understand the common model organisms in molecular biology. First, E. coli. This E. coli is a prokaryotic model organism to study the gene expression, DNA replication, transcription, and translation process. Okay, and this E. coli plays a most important role in research area of genetic molecular genetics. Okay, and this E. coli is easy to grow and genetically manipulate. Okay, and the lifespan of E. coli is very short, and the genome size of E. coli is also short. And this E. coli this E. coli is used in rDNA technology. We use this E. coli in rDNA technology to produce for production of proteins, enzyme, secondary metabolites, etc. Okay, and to see the gene expression of a particular gene. Okay, next is yeast. Yeast is Saccharomyces cerevisiae. This is also known as a baker's yeast. It is a model organism of unicellular eukaryotics, and it is used to study the cell cycle regulation, genetic and metabolisms. We study this E. coli to understand the different catabolic as well as anabolic pathways. Okay, and this yeast olfangei shares a many fundamental cellular processes with higher eukaryotics. Next is a Saccharomyces pombe. This is also known as a fission yeast, and this E. coli is used to study the cell division or chromosomal dynamics. Okay, first is mhm, first is Saccharomyces cerevisiae. Saccharomyces cerevisiae is also known as a baker's yeast, and Saccharomyces cerevisiae is a model organism of unicellular eukaryotic cells, and this is used to study the cell cycle regulation, genetics and metabolism. This E. coli is also used to understand the different catabolic as well as anabolic pathway or metabolism of any organisms. Okay, next is the Saccharomyces pombe. This is also known as a fission yeast, and this is used to study the cell division and chromosomal dynamics. Okay, next model organisms is C. elegans, and this C. elegans is a multicellular organism, and this C. elegans is used to understand the development of the cell or development of organisms, neurobiological processes, and aging. And by the help of the C. elegans, we also study the cell lineage, or we want to use the C. elegans to see the feature of the cell and well characterized genome. Okay, next is the Drosophila. This Drosophila is a classical model organism for genetics and developmental biology, and it is also share a higher percentages of gene with humans. The genome of Drosophila have a higher percentage of similarity with the genome of humans. That's why this Drosophila is used to study the gene function, inheritance, and evolution pattern. Okay, next is the zebrafish. Zebrafish is a vertebrate model organism, and this is used to study the development, genetics, and diseases. And transparent embryo allow the real-time observation of development. The embryo of zebrafish is transparent. That's why this is used to study the real-time observation and development in embryos. Okay. Next model organism is mouse. This mouse model is a model organism of mammalian model system, or this is a model organism of human. Okay, this mouse is widely used in biomedical research, genetics, immunology, rDNA technology, etc. Okay, and this mouse is genetically similar to humans. That's why it is plays a most important role, or this is very useful during the drug discovery and disease modeling. Okay, the transgenic mice and knockout mice are widely used to study the gene function. Okay, suppose we want to know the expression of gene particular gene of human beings, then we inject this gene in mouse. Now this mouse is transgenic mouse. Now we study this gene expression in mouse. Suppose this is a particular gene X, and that is present in human beings. Okay, now we want to see the gene expression of particular this gene X and the role of this gene in biological process. Okay, so to see the gene expression of this particular gene, we use a model organisms or mouse. Why we use this mouse? Because the genetical conformation of mouse is very similar to humans. Okay, how we perform this process? Suppose, okay, how we perform this process? First, we prepare our gene of interest, then we enter this GOI in mouse, and whenever we inject this GUI in mouse, by the help of our DNA technology, then we see the expression of this particular gene in mouse. Okay, suppose this suppose this particular gene code a protein, that is, suppose this particular gene express a protein, that is P, and this plays a most important role during the digestion. Digestion in digestion process or digestion system in mouse. Okay, then this particular gene X also express this particular protein P in humans and also play the most important role in and also participate in digestion system of human beings. The expression of this mouse is the expression of this particular gene X is similar in mouse or humans. In mouse, this gene suppose in mouse this particular gene code a protein that are participate in digestion system. Same gene expressed in humans also code a protein which participate in digestion system of humans. Okay. 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 To understand in this way we use a model organisms to study the different biological process and the function of proteins, the function of enzyme and function of secondary metabolites, etc. And the expression of gene in model organisms is very similar to the expression system in complex organisms or humans. These are the animal model organisms. Okay, now these are the plants model organisms. Now Arabidopsis thaliana is the model organisms of plant. This is a small problem plant used in plant genetics and molecular biology. Okay, to study the biological process of biological process biological process of plants. Biological process of plants. Okay, and the size of genome is why we use this plant as a model organism, because the genome size of this plant is small and the life cycle of this plant is also small. Okay. Mhm. In case of plant, Mendel hmm, in case of plant, Mendel's experiment on pea plant is also a example of model organisms or model plant. The rule of the rule of the the three rules of genetics. First is law of dominance, law of independent assortment, law of segregation. These three laws also applicable in most of the plants. And these three rules are studied on a single pea plant, but this three rules is applicable most of the plant. This is also an example of model organisms. Here we use this pea plant as a model organisms. We study the three rules on pea plant and these rules are applicable most of the plant. Okay, and during the during the law of inheritance during the law of inheritance we use pea plant as a model organisms as a model plant. Okay, and this law of inheritance is law of dominance, law of independent assortment, and law of segregation. Okay, next we understand the characteristic of good. Okay, first we see the E. coli, and E. coli is the model organisms of bacteria. Okay, we first we study the E. coli, and E. coli is a model organisms of bacteria, and this is used to study the gene expression, central dogma, and molecular genetics. This E. coli plays the most important role in rDNA technology to understand the for the production of protein, enzymes, and other metabolites, and to also study the gene expression system. Okay, next is the Saccharomyces cerevisiae. Saccharomyces cerevisiae is a model organism of fungus, and this is used to study the cell cycle regulation, genetics, and metabolism of higher organisms. Okay. Next is the C. elegans. C. elegans is a model organism of round worm, and this is used to study the and this C. elegans plays the most important role in developmental biology. And by the help of C. elegans, we study the cell lineages or the feature of the cell. Okay, next is the Drosophila. Drosophila is a model organisms, and the Drosophila is a classical model organisms for genetics, and also play the most important role in developmental biology to understand the gene expression, function, and evolution pattern. Okay, next is the zebrafish. Zebrafish is a model organisms for vertebrate, and the transparent embryo of zebrafish is used to this zebrafish is the model organisms of vertebrates, and the transparent embryo of zebrafish is used to study the real time process and observations. Next is the next is the mouse. Next is a mouse. Next is a mouse. Mouse is a model organisms of mammalian. This mouse is plays a most important role in drug discovery. Next is the Arabidosis thaliana. This Arabidosis thaliana is a model organisms of plant. Next is the Pisum sativum. Pisum sativum during his Mendelian use a pea plant during his experiment to understand the inheritance pattern in plants. Now we study the characteristic of good model organisms. First, easy to grow, easy to maintain in laboratories, short life cycle, well characterized genome, amenable to genetic manipulation, and relatively simpler structures. These are the characteristics of good model organisms. Now we study the selection of model organisms. First is research consideration. First, first is first is research consideration. First, relevance. Okay, we choose a model organisms according to our research work. Suppose we want to study the drug. Study the we want to study the drug. We discover a drug. Okay, for humans. Now for humans. Okay, now In this case, we choose mammalian model system, and the model, and in this case we use a mammalian model organisms. Mammalian, and the example of mammalian model organisms is mouse. Okay, in this case we prefer mouse rather than other model organisms such as yeast, E. coli, C. elegans, Drosophila, zebrafish, etc. We choose our model organisms according to our research work. Okay, suppose we want to study the cell regulation pattern. In this case, we use a yeast as a model organisms rather than mouse. Okay, we choose the model organisms according to our research work or our how, okay, research work, and it is also depend which types of biological process we want to study. Next, conservation. There are lots of biological process or biological functions that are conserved. In this case, we use a model organism to understand the pathway of conserved gene. Okay. Next, next is particular consideration. First, easy to maintain. The organisms or the model organisms should be easy to maintain or grow in our laboratory. In laboratory, there are lots of factors which play the most important role. First is the space requirement. Okay, how much space is needed for growing or for breeding the model organisms? This is very important. Next is diet. Okay, which types of diet is required for model organisms? Next, lifespan. And the lifespan of model organisms is very short for faster experiment and observation. Okay. Next is genetic manipulation. The ability to manipulate the organism's genome is highly desirable because to perform a lots of molecular techniques. First is gene editing techniques. Next, first technique is gene editing. In CRISPR technology, gene we modify the genome of a model organisms to study the gene expression or to study the function of a particular gene. Next is transgenic. We also introduce a foreign gene into a model organisms to see the gene expression and for production of proteins, metabolites, enzymes, etc. Next, mutagenesis. We also mutated the genome of model organisms to study the disease or gene expression system. Next, ability to Next, availability of resources. The chosen organisms should be well established in research community, and this required existing research tools and reagents, and these reagents and tools are antibiotic, plasmids, and proper established protocol. Next, shared resources, and these shared resources are the core facilities for imaging the model organisms, for sequencing the genome of model organisms, and other techniques such as microscopic techniques. Next is a wealth of published data. This published data provide a foundation of our research. Okay. Next is the ethical consideration. First is the animal welfare. Whenever we use animal as a model organisms, in this case we should strictly follow the all ethical rules and guidelines. Next is the alternative. Next is the alternative. Okay. Whenever possibly, research should consider using a non-animal model. And the example of non-animal models are non-animal models such as cell culture, computer simulations to reduce the use of animal in research field. These are the criteria which we follow during the selection of model organism. First is a research consideration, practical consideration, genetic manipulation, availability of resources, and ethical considerations. These are the selection criteria of. Hmm. Now we study the limitation of model organism. First is the specific to the model. Finding a one, Sometimes finding a one model organisms may not applicable to other organisms even within a same taxonomy group. Next is the oversimplified. Sometimes model organisms often represent a simplified system as compared to complexity of human biology, and this can lead to oversimplified view of biological processes. Next, Next is the genetic manipulation. Genetic manipulation in model organisms can have unintended consequences potential affected to other biological processes. Next is environmental impact. Releasing the modified organisms into the environment can have unpredictable ecological consequences. We discuss the addressing addressing the limitations. First method is a refinement of matter. Refinement of model. Next is the development of alternative models. Next is the multimodal approaches. Next is the ethical consideration. Refinement of models. Ongoing research aim to develop a more sophisticated and human relevant model organisms, and such as genetically engineered might more accurately mimic the human diseases. Next is the development of alternative models. Investigating and refine the alternative models such as our organ on a chip technology, human-derived cell culture, computational biology can reduce the dependency on animal models. Next, multiple model approaches. Utilize a multiple model organisms and integrated the data from different systems can provide a more comprehensive understanding the biological process. Next, ethical consideration. Next is the ethical consideration. Prioritizing ethical principle and minimize the animal use in research field is crucial. That's why presently we generate alternative methods, and these alternative methods such as computational biology and cell culture. Okay, this is all about today's lecture. Please follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, and also get it on Google Play Store. Thank you so much for watching my video. Please like, share, and subscribe ITLS Academy. Thank you.